last year I was like actually doing Alzheimer's disease, and this year I'm doing genotyping of mice at Columbia University. My mentor's name is Dr. Basiru-Lee-Leigh, and the actual mentor I was with for like the whole summer was Lily too. And I had Maria Perez, Dr. Aruna, and Harold Bruno. Just to introduce you, like genotyping. Genotyping is like a genetic foundation of the cell, an individual organism. Genotyping is the process of determining the genotype of an individual or organism. The current methods of genotyping include PCR, DNA sequencing, ASO probes, and hybridization. I don't know what they actually are, but yeah. The reason we use mice is because we actually like have the same kind of like structure, like bones, brains, and like how we act. And plus, we have like 3.1 million of the same base genes, basically chemical formulas. And actually they're affordable and cheap, and kind of like strong in them, so we use them. The purpose of this research was to determine whether a specific gene is contained in a mouse or not. That's like the general one. And in our case, we're looking for a gene called CRE, which is a man-made gene. And what it basically does is like a knockout gene. If it is inserted in mice, all it does is like take out a gene that isn't wanted. Okay. Yeah, see this place, right? This place we actually have to do all the work because like we don't want anything to contaminate it, so we actually have to do everything inside it. This actually is the model we cannot use the temperature model because it had already been done. So these babies we have to use, they are like 21 years old, so you actually have to cut up for like 1.5 inch from the tail and use it for the experiment for the DNA isolation. You're not practice now, all you have to write is DNA isolation. Alright, the first thing we do is like cut off the tail and put it in like a tube, it's kind of clear, and like we add protein and proteins, K and lactic buffer, so you can like separate the tail, separate this tail into like small pieces, and like just leave like the tissue inside. That's what he said. After that, we centrifuge for like 3 minutes, and this is what happens. All the hair of the mice tail just goes to the corner of the tube. And then next, we take out the liquid, since that's what we need. Put in a new tube, and then kind of like centrifuge it again, so we get our oil protein out. After centrifuge, it's going to be like a big white spot, like right here, which is the protein. This one we don't need it. So what we do is that we take out all the liquid, leaving the protein inside. And we centrifuge it again, just like to make sure that all the protein is gone. You have like a little bit left. And we take out all the liquid again. Take all the liquid again and put it in a new tube. After you like centrifuge it again, it comes out. It's the last centrifuge, I know. After we centrifuge it again, you'll see like a small black pellet in the end, which is like right here. That's the DNA sample we need. After we add we actually wash the DNA sample using ethanol 7%. We actually wash just like everything around it, and we centrifuge it again, and add up all the liquid. Then put in another tube, the same tube, but use another liquid, which is actually distilled water, and then we wait for like an hour or two. This actually the gel is like the easiest thing to do. I actually made this one. What we do is we mix the powder, and then heat the sample for like 2 to 3 minutes. Then add ethyl bromide. For this one, we actually have to be really careful because if we inhale it, you could actually have cancer. So we have to be really careful. And we have to pour the solution in mold with combs and then leave it at room temperature. Sometimes we leave it there for like 20 minutes, or sometimes up to like 3 to 4 days since like it never melts. for the PCR to work, right? We needed to use the buffer and the DNTP, the primers, which we used like 3 primers. We didn't need the DTA essence, we only need to put the 3. So we used 3 primers and use the DNA sample, the tag, and the DMSO, and also put the water since we needed to. And that's basically how the PCR works. It denatures anneals and extracts. This is the actual PCR machine used. This way we actually put the tubes in and then let it run for like 35 circles, which will take like 6-7 hours. Okay. After we're done with the sample, we actually leave it for like an hour after the sample is done. The first step we do is that in aqueous gel, after it's solidified, we inject the samples inside the second and third one. Keep going from the second all the way. The first one is always DNA ladder, which actually tell you where the DNA is actually working, like this one. It works like this one. You can actually tell where the DNA is actually at. After we're done with the samples, we actually take it to a machine and I just show you what the machine looks like. This way with the machine, this is the UV light, and this is like a camera that takes the picture. And this way the picture comes from. It's like a TV, and after we take the picture, we have to take the thing back to our mentor or our lane to look at the pictures. And for our results, we found out that 4 of the mice, these 4 actually had 3 out of the 9. And the other 9 mice, since we didn't need them, they have to be killed. And for these ones, we use them for like tissue culture. Okay, in conclusion, 3 of the mice, 4. I will take this page. 4 of the mice contain the CRE gene, and like for now, we are now trying to find the DTA gene and the FOXO3 gene, which we need for tissue culture. And that's my future work to learn how to do tissue culture next year, maybe after school. And that's my references. I would like to say Dr. Staley, Dr. Denizou, Dr. Aruna, Marie Torres, Harold Bloom, my parents, Dr. Manavali, Columbia University, Dr. Stad, ACSO staff, and everyone else.
Why did you need to know whether the mice contained CRE in the first place?
Because my mentor is doing like another research, which is tissue culture, because she needs the CRE gene, which actually knocks out all the genes that they don't need. Some of them might have like different kind of genes in them, so if we don't need them, everything, so the CRE gene inside of them, so the other genes will be taken out. That's why we need the mice with CRE genes.
Why do they have to be 21 years old? I mean 21 days old.
Because that's when they start like breastfeeding from their parents. They can't do it because I get to rules, I guess. We can't do it before 21 days old. Any other questions?